Hello, everyone. Good morning, good afternoon, good evening. Joining us from other parts of the world. Today, it is with great pleasure I get to introduce my friend and colleague, Dr. Ian Villamagna, who'll be giving us a talk. He'll be discussing our novel ionizable lipid portfolio for enabling non-viral delivery. Just a reminder of our team. We have Kim Killian as our regional sales manager located in Southwest USA in San Diego area. Ian in the Northwest as a senior field application scientist there, and myself and Ingram in the Southwest region. And all of these events, as well as our previous events, are recorded and are on our YouTube website, where you can go back and look at all the old events, including this one. They will all be archived on the playlist linked here. And I think without further ado, I'll hand it over to you, Ian.
Awesome. Yeah, thank you, Via. I appreciate it. Appreciate the introduction, and we appreciate everyone making it this morning. So we wanted to kind of host a webinar to talk a little bit more about lipids. This has been a really strong focus for Precision Nanosystems within the past year. I think a lot of people know us as the instrumentation company, which is still very much true. But we've been doing a lot of work in the lipid space as well. Very exciting work, and I think work that really deserves to be talked about and to be shown to the world as much as possible. So the goal for today is to kind of give some background and insight as to what we've done in the novel ionizable lipid space, kind of show some data, show kind of what our lipid library looks like and how it functions, and really set the stage for any future conversations that anyone may like to have about how you can work with us in the lipid space. So it's not really any secret anymore or any surprise that Precision Nanosystems really launched very heavily really a couple of years ago into this genomic medicine space. So right around the start of the pandemic, but even right before that, I would say, we really saw this area of genomic medicines as the key to kind of the future of medicine. So looking at the ability to take different types of nucleic acid, whether it's something like a more traditional siRNA or self-amplifying RNA, CRISPR-Cas components, and really have a strong control over disease with those types of modalities. We saw that as really the future of medicine. And we saw ourselves uniquely positioned to be in a place where we could be a technology provider for groups that are developing these types of therapeutics. What we found as we've continued, and what we really continue to see, is that these types of therapeutics that are being developed really require a very different type of technological expertise and really different types of technologies along the way. And that's really at the heart of everything we're doing, both on a manufacturing perspective for nanoparticles, but also in a lipid perspective as well. And so we'll kind of go into that today and talk about really the differences in being a technology provider and a true partner with our customers, and show really what that looks like. For us, and kind of going back to that thought about having a more complete sort of picture of technology, genomic medicine really involves every aspect that you're seeing on the screen here. So it involves that nucleic acid payload, all the way really from design up to manufacture of that payload. It also includes the lipid components to make the drug product itself, and all of the different lipid components that you see on the screen here, or other types of components to make particles as well. Something like polymers is still very relevant in the field. We feel that in order to really effectively develop these therapeutics, it's really beneficial to have a technology provider and a partner that has the ability to really bring all of that expertise and all of those components of technology together under one roof. It enables a quicker sort of acceleration into developing these therapeutics, which really enables different types of applications. And so on the right-hand side of the screen here, you can see some of the routes that our customers have taken right now. But obviously, as you can imagine, we have a very wide range of customer base that is working in multiple different types of applications within the genomic medicine space. The area in and of itself, and just by nature of what we're doing today with these types of therapeutics, is inherently challenging. Nucleic acid delivery has never been overly straightforward, and I think we can see that dating back 10, 15, 20 years even now in publications where it was apparent very quickly that especially for things like RNA, it really required a more complex sort of delivery system than just taking that drug product and putting it into a living body. It requires that really complex delivery perspective as well. With that, the complexity that comes along with that is just simply that those delivery systems are not inherently straightforward either. Traditionally, there had been a knowledge gap in the space. So nanoparticles were never necessarily a massive topic where you could find thousands of experts in the area. The ability to take what you were doing at a preclinical scale and then move it into a clinical development phase was unclear and uncertain. And really, what we've, I think, seen more and more recently is that different programs have very different needs. So different RNAs, different payloads, different nucleic acids, they all have slightly different needs in terms of developing delivery systems and developing genomic medicines. And so that requires a level of flexibility in order to really effectively develop and really deliver these types of medications. So we really believe that the pillars of a genomic medicine and the pillars of a successful genomic medicine come down to these four main components. So first and foremost, having a specific target for your indication is really the first key that a lot of our customers bring to us quite early, and that's something that has been elucidated quite early on in the research phase. From that point in time, you move into an area that Precision Nanosystems has capabilities to assist with that technology. So whether it's the payload and making sure that we have a very effective payload for that disease target, whether it's the nanoparticle itself, so the delivery aspect with the lipids or any other components to actually package that payload and bring it into a cell. And then a manufacturing platform that really has true scalability to go along with it, such that we can work with confidence at a preclinical scale, knowing that it will function and will move to the clinical scale is really, really important as well. In order to allow for this to happen, Precision Nanosystems has built this genomic medicine toolkit to enable that. And so what we've done now is developed multiple different platform technologies in order to really hit all of those key pillars that we feel are very important for a successful genomic medicine. So we have a very nascent genomic payload platform where we're looking to develop self-amplifying RNA for customers that bring a disease target to us. We have our Genvoy delivery platform, which is our lipid-based platform, which we'll talk about at length today, but really looking at leveraging our understanding of lipids, different ionizable lipids, different combinations of lipids into making delivery systems that are much more effective for specific types of applications. And then finally, still true to our roots, our manufacturing platform, which still offers really unparalleled scalable nature for nanoparticle production across the board. This is just one component of developing that genomic medicine, and we understand that. And it's not insignificant. It's not a small portion of that process, but it is one part of that process. With our acquisition by Danaher and now being part of this Danaher Life Sciences family, what we're able to do now is really leverage our partners within the greater organization. So really working with our colleagues at Cytiva and Paul for things like downstream processing, working with our partners at Aldevron, IDT for upstream type of applications, and then really having the ability to look at other places as well within this organization, Beckman Coulter or SciEx for other applications like analytics, for instance. We're really able to look at this and build this broad sort of portfolio of what we have to offer, and really believe that we are as close to a one-stop shop for this type of medication development as you'll find on the market today. So that's just some background. To kind of get more into the lipids and the science here, what we're going to kind of look to go through today, first and foremost, is actually just a little bit of a background and reminder on the manufacturing platform, which is inherently tied to the lipids and is quite important to kind of keep in mind throughout this. We will talk about the concept of our lipid library, kind of what it is and how it functions. We'll give some data on some of these next generation genomic medicines, so some vaccine data, some gene therapy data. And then we'll kind of touch on some of the challenges and how the technology looks to assist with that as well. So before we jump into lipids specifically, I do want to just have a reminder, if you will, about the manufacturing platform, because it is really key to what Precision Nanosystems does, and it really is historically who we are as a company, and still remains to be very important for us. Optimal nanoparticle assembly is achieved through bottom-up assembly, so allowing these particles to self-assemble through some type of interaction between two fluid streams is really what we look to provide in order to effectively formulate nanoparticles in an extremely homogeneous fashion, so having a really high level of quality across the board. What you're looking at here on the screen is that process happening through an artistic rendering. And so what we see, first and foremost, are the components mixing together in their fluid streams. This happens through our microfluidic architecture, which you see on the right-hand side of the screen. And as those two fluid streams, an aqueous and an organic fluid stream, move through that architecture, what you get first and foremost is an interaction between the ionizable lipid in light blue and the genomic payload, in this case, RNA or DNA, in green. That initial interaction is really key to the formation of that particle and really begins the formation of that greater particle itself before other lipids, such as helper lipids and cholesterol, which are shown here in purple and yellow respectively, come in to kind of fill the void, and then finally a stabilizing lipid added in for stabilization purposes. All of this process happens through the microfluidic architecture that we have on the screen here. And that architecture is really, really key to what we do. It is the differentiator between Precision Nanosystems and other types of technologies in the field. What this architecture really looks to do is take that mixing process of an aqueous and an organic fluid stream, where that nanoparticle preparation is happening, and do that in the most reproducible and homogeneous way fashion. So with what we have here, we're able to add those two fluid streams together and have a very homogeneous mixing of those fluid streams, which leads to a very homogeneous mixing and production of the nanoparticles themselves. The other nature of that architecture, and the other real key to it, is that we've been able to develop this architecture to create that mixing without any sort of 3D structures within it. And what that enables us to do is very simply use the geometric aspect of it to make it larger, and allows us to really have a true platform that can scale up effectively. The exact same architecture that we're using at a clinical manufacturing scale is the same thing we're using at a preclinical scale, because of that architecture. And what that allows us to do is work very confidently that with knowing that the particle preparation you're doing at a preclinical scale is the same as what will happen at a clinical scale. That architecture in and of itself that you see on now the left-hand side of the screen here is really key, and really is what has enabled us to have a manufacturing platform that can go all the way from an early discovery stage, where we make under 1 mil of nanoparticle solution, so up to 250 microliters. But then taking that exact same architecture and using it across the board through early preclinical development with Ignite and Ignite Plus, through process development and late preclinical studies with Blaze, and then into clinical and commercial manufacture as well. Regardless of where you're working in this space, the instrumentation changes, but the microfluidics do not. And so you can work very confidently knowing anything you're doing on a day-to-day basis on Ignite has the ability to be scaled up into the clinic through this process. And we've seen this multiple times, and we've shown it multiple times. This is actually, I would say, an older example that we still utilize because it does show quite well what that is. But we've seen this again for different types of applications, including vaccine production, production of gene therapy, therapeutics, and things of that nature. But what this really goes to show is that the particles you're making, in terms of particle characteristics, so hydrodynamic diameter, the dispersity of the population, and then the encapsulation of the payload, really doesn't change regardless of whether you're working on our preclinical system, the Ignite, at 12 milliliters per minute, up to Blaze for process development at 60 or 90 mils per minute, and then into GMP at a clinical scale for 200 mils per minute. The particles that you're making are the same, and we do have good data as well showing that the biological efficacy of these particles is retained across the board. So there's no difference in the particle formation at a large scale versus a small scale. To give kind of the overarching example of where we sit within the organization, so we really work in more of the upstream space right now, if you will, kind of after your payload for the most part. So we partner with Aldevron, Cytiva, IDT for sort of true upstream development. Our instrumentation sits in the middle, so you can imagine you have your payload. We go ahead and work with the lipids in the encapsulation space to get it into a delivery system that's effective. And then now we have the power of Cytiva for downstream processing, things like tangential flow filtration, sterile filtration, and fill finish as well. So being able to go all the way from start to finish, again, within that Danaher family, and with logical technological solutions along the way. All right, and so shifting gears a little bit here into lipids and our lipid library that we have. This is a newer platform that we have been developing. The thought process for developing it is not new, and we've seen this for quite some time. I would imagine for most people that are here today, I don't necessarily have to go into the complexity of the lipid space today, because I think we see it on a near daily basis, especially for those of us in the field. It's a constant reminder of kind of the IP issues around that lipid space and kind of who's fighting who, if you will. What we have looked to do with our Genvoy delivery platform is, first and foremost, remove ourselves from that initial sort of group of lipids that have those IP issues. And then secondary to that, we really see the lipid space as an open space where people need lipids, and people need lipids that work effectively at an early stage of development. So what we're really looking to do with this platform is democratize the ability for people to work with our lipids at an earlier stage in development, knowing that they have the ability to bring that lipid and that delivery system that they develop all the way through to clinical manufacture. That idea of democratizing the ability to do this to as many people as possible is really inherent to what P&I is and always was. That was our initial intention with the manufacturing platform, and it's really continued and been brought over with the lipid platform as well. The way that we've gone about doing this is through the development of a lipid library. And I think that it is important to understand when we say library, what that means and how we really think about it. What we've been able to do is develop, first and foremost, a library of over 120 ionizable lipids that are proprietary to Precision Nanosystems. So our team in Vancouver went through and developed all these different lipids with different combinations as well. And so that's the second aspect to it, is we have 120 ionizable lipids, but we also have the ability to mix those with different combinations of helper lipid, cholesterol, stabilizing lipids, so on and so forth, in order to create a really broad range of ability from our lipid library. It's something that we continue to innovate in, and so we're always adding new lipids. We work with our customers to develop lipids that are more developed for their purpose rather than sort of as a generic lipid mix, and it's something that's being constantly refined. The way that we go about grouping our lipid library is by application, and how it essentially works for us is that we've gone through and developed these lipids, and we know through testing internally that we have a subset of this library that works extremely well for something like a cell therapy application. So ex vivo translation of T cells, for instance. We have a subset of those lipids that we know work extremely well for a vaccine application, and then another subset that we know work very well for traditional gene therapy applications. And that's how we've begun to build this library out with these larger areas that we think are logical places for our customers to start working, that we can always narrow down from there and really look to select a lead candidate. So we'll show some examples of that today. We'll go through these 3 different examples of 3 of the areas that we work in right now, starting with vaccines, gene therapy, and then cell therapy as well. The vaccine space obviously came by nature of the pandemic, and a lot of the work we did in the vaccine space with this lipid library was done in response to creating a COVID vaccine on behalf of the Canadian government as part of a pandemic preparedness model for that government. So we look to do all of our work in the vaccine space with self-amplifying RNA, which we believed was a really unique sort of application that allowed for a much lower dose, much easier sort of development process to get to that point because of that dosing regimen, but still allow for a very potent immune response and a very effective vaccine as well. A lot of our studies with respect to our vaccine work look very similar to what our customers would be doing and probably look very similar to what you have come to notice and expect from groups like Pfizer, BioNTech, Moderna, things like that, where they're developing vaccines that are on the market today. So for one example here, we looked at in vivo testing of our lipid nanoparticles with the self-amplifying RNA. First in a rodent model, dosing as a prime boost, so on 0 and 4 weeks, and then looking at IgG antibodies and also the response from the spleen as well. And we're able to see here from 2 of our lead candidates, so LNP1 and LNP2, that both LNP1 and LNP2 did have a very efficient induction of the SARS-CoV-2 IgG response in these mice. We did see that one of these candidates had slightly higher activity as compared to the other, and that was LNP1, which in this case, we were looking to develop a lead candidate from those 2 different lipid mixes. But both LNP1 and LNP2 showed neutralizing antibodies against the virus as well. So an effective vaccine from an animal perspective in rodents. And that allowed us to move into a non-human primate study phase, where we were able to look in a large animal model with primates, looking again at day 0 and day 56 for a prime boost schedule. And this really followed along with what we kind of expected from a prime boost model. So looking at a significant difference in IgG levels at day 42 compared to pre-bleed, showing that the vaccine was effective in that prime phase, and then the response at day 70 after a boost, again, much higher than earlier time points, showing that the traditional prime boost was effective with these vaccines. To give more of an indication as to how the ionizable lipid library played into this work, the data you saw in the previous slides came down to 3 different vaccine candidates. But in order to get to that point, we started with the candidates that you're seeing along the bottom of the x-axis here for our vaccine work. So these ionizable lipids that we have listed along the bottom here really are the lipids that we believe work effectively for a vaccine application. And it shows really that we believe we have a vast number of lipids that work really effectively in that vaccine application. So everything along the bottom that you see are different lipids that we tried in formulation with the self-amplifying RNA. We did do comparisons to SM102, which is sort of the standard state-of-the-art lipid right now for vaccine application. And we do see that we have similar results across the board with our vaccine lipids that we have. What this means for our customers, realistically, is that we have a really broad range of ionizable lipids that we can begin with. And so you'll remember from earlier when I was talking about sort of minor differences between formulations that do require changes. What this means is that we have a really broad umbrella that we can cast with customers to begin that development work much earlier. So we're able to look at a vast range of ionizable lipids and find one that works effectively for our customers, rather than having sort of a answer solution where we're trying to force one specific lipid into that space. We have flexibility to find one that works most effectively for a specific indication. As a second example, and maybe a more traditional example of where our lipids sit, gene therapy is one area that we continue to look at and continue to really see as a large area for us and for these lipids. So we'll give an example today of some work that was done in an anemia model. So looking at EPO therapy, so the production of EPO from mRNA. And in this case, again, what we were able to see is that with this type of therapeutic, you're really looking to deliver, and it will be tracked to the liver. We're able to see again that we have a very broad range of ionizable lipids that did show a high level of efficacy for this type of application. Some, in this case, I would say even more realistic than others, especially as you see towards the right-hand side or downfield of that graph that we have. We're able to start seeing lipids that we believe would be more effective in that space for the payload that we're using. Much like the last study you saw, we did compare this to sort of industry gold standard, which is MC3 in this case, which is your standard sort of ionizable lipid mix that has been used quite readily across the board since 2018 or even before that, and showing that we get good efficacy in comparison to that sort of gold standard lipid as well. But again, in this case, lots of different applications, lots of different places for our customers to begin in a gene therapy space that we feel would be effective for developing these types of therapeutics. Perhaps more interestingly, I think, is sort of the approach that we've seen with gene editing. And I think this is interesting because you can see right away on that graph on the right-hand side that we've now taken this library that for something like a gene therapy application, which is slightly more standardized, we've now taken that library and really whittled it down into a much smaller subset, where now we're working with roughly 6 different ionizable lipids from that library. But we see that these 6 ionizable lipids work really effectively for a gene editing perspective. So something like a co-encapsulation, a Cas9 mRNA, and then guide RNA as well. So in this case, we're looking at the TTR CRISPR gene editing, and we're looking at TTR percentage after delivery, where we see a strong reduction from these lipids and from these therapeutics that were developed with these lipids across the board. Up to a 75% reduction in the TTR protein levels were achieved after 7 days with just one dose of this therapeutic. And so what this is really showing is that we're able to really take that library and bring it down to a stage where we can find things that work effectively for different applications. So in this case, the gene editing application we're showing here, but then also different types of applications in this space, like base editing as well. In this case, looking at PCSK9, which has been shown to lower cholesterol levels. And again, we can see that with different lipids as well and different sorts of payloads, we're really able to get into a space where we've developed lipids that have the ability to really have a more effective nature on what the overall target is. So what we're looking at here is actually both a single and a double dose, and looking at some different sort of editors that were synthesized, all of which are still being encapsulated in those lipids, which we've seen to be quite effective for this type of application. This is something that gets asked about quite frequently, and so I do think it is quite important. The tolerability of the lipids that we've seen, and this is true really across the range of our lipids as far as we've seen, but in one example, so using ionizable lipid 2 as its denotation from PNI, we did look at comparing that to the 2 lipids that are in the vaccines from Pfizer, BioNTech, and Moderna, respectively, so SM102, ALC0315, and really looked at comparing them from a tolerability perspective. And what we see, and this is really what we see from the lipids within the library at large, is that they are tolerated extremely well. So in terms of clinical observations, but also things like necroscopy and the liver histology, we see that the PNI lipids are tolerated extremely well, and we don't have any noticeable side effects from our animals when they're treated with that. All of this was done in a blinded study by a third party. And so we're able to move forward with a high level of confidence with these lipids that they are going to be not only effective, but safe as well. And then finally, so we'll kind of close our lipid talk here. And I think it's fitting because in my opinion, I think this is one of the most interesting applications that we've been working on and is really interesting to kind of see the field grow very rapidly right before our eyes. Precision Nanosystems launched into the cell therapy space about 1.5 years ago, 2 years ago, maybe now, with the understanding that we had learned a lot from in vivo applications, and we believed we could take that understanding that we gained and apply that into a cell therapy space. So apply that into a more ex vivo space as well. So looking at isolated T cells, treating those T cells with lipid nanoparticles instead of conventional methods like electroporation or viral vectors, and really develop T cells that can be used as drugs by nature of using lipid nanoparticles. What this really comes down to in terms of using lipid nanoparticles for this type of cell engineering is the ability to really have a complex engineering of your T cell population. So traditionally with methods to transfect T cells, you do take a hit to cell viability. And even if that viability comes back, which many times it does, what we see is that after that initial hit, you have a cell population that is not as healthy and doesn't respond as well down the line. Much like our lipids are tolerated for animal studies, we see that with cell populations, and T cells specifically in this case, the lipid nanoparticles are tolerated extremely effectively to the point where we don't see at relevant doses hits in that cell viability really at all. So nothing below the 90% range for the most part. And when we do that, what it enables is a very healthy cell population across the board. And that enables us to do really sort of complex engineering with our T cells. So we can do standard gene delivery with lipid nanoparticles. We can have T cells express CD19 CAR constructs. We can also do knockouts with them. So we can knock out different receptors on that cell surface, either single or double, like you see along the bottom here, where we did a TCR and a CD52 knockout. And then maybe giving an indication as sort of the complexity that can be done. You can get all the way to doing T cell development where you have knocked out different receptors and then also express something as well. So in this case, a TCR knockout and then a subsequent CD19 CAR expression on that surface. Multi-step engineering of these T cells is really the key here. And that's something that we believe very firmly that lipid nanoparticles enable and allow us to do effectively and is really going to change the way that we can develop T cell therapies in the field. The data for this type of application is included in this application note on the screen here. And I do recommend reading it. It is on our website. Feel free to reach out to us. We'd be happy to get it out to you and then discuss it at any point. It's a very interesting data set that I think is quite eye-opening as to what can be done in that T cell space. As you would expect from Precision Nanosystems, this T cell therapy technology does scale as well. So we have developed a lipid mix that is specific to T cell populations right now. We sell that in the sense of a T cell kit. So an off-the-shelf kit that enables development of cell therapies from an early preclinical stage. And what we saw very rapidly is that there was an appetite to work with larger cell numbers. So we took that from our discovery stage and then scaled it to the true preclinical stage with the Ignite. As you expect, and as you've come to expect from every technology that comes from Precision Nanosystems, we've seen that scalability continue here as well. So both in terms of particle size, so particle characterization, the amount that you can encapsulate, and then also the biological activity of it, cell viability, CAR expression. We do see a continual scalability from these types of lipids in the T cell space as well. And we have every indication here that we're able to get from discovery to preclinical scale with this lipid mix for T cells, but then also go beyond that as well. So again, both from a particle characteristic but biological perspective, we feel very strongly that we are able to use these lipid nanoparticles with the microfluidic platform for a really continuous production of these therapies, and then eventually incorporating large-scale manufacturing to get these types of therapies to the clinic effectively. So I'll just close with kind of how we work in the lipid space for everyone. First and foremost, we do have research use only kits that are available. Genvoy ILM is a research use only lipid mix that is used abundantly in the field for any number of applications. And so it has really sort of ubiquitous nature as to what it can be used for and where it's being used. When customers feel it's the right time, and that's something that, that conversation will happen as part of a natural sort of partnership with your local team from Precision Nanosystems, your application scientists, and your sales managers. We can begin to look into the license and partnering model as well, which typically begins with a research evaluation agreement. So taking a look at what you're working on, looking for lipids that we think would be effective for that application, and then testing them to find a lead candidate. After that point, we look to move into more of a licensing agreement as well with that lipid to really enable you to go from that preclinical stage into the clinical development process. Anyone that has questions about this, we're more than happy to have just a really sort of high-level initial conversation about what that model looks like and how we feel it looks to get from sort of early preclinical to late preclinical, and then eventually into clinical as well. And we do have examples of this, and so this is a very common question is, all of this data is lovely and great, but is this actually being used clinically? We do have publicly disclosed partnerships on our website as well with two groups that you see here that are looking to develop these genomic medicines with our lipid library, and so then with our manufacturing platform as well. There are more examples of this that are not publicly disclosed, of course, but these two are two publicly disclosed examples that we feel very strongly about and feel are a very good indication of what can be done with our lipid mixes. So really our goal at Precision Nanosystems and our continued goal is to accelerate the creation of transformative medicines that significantly impact human well-being. And we really feel that by adding this very robust lipid library to this space, we're really staying true to that mission and goal. We're looking at different types of areas, whether it's vaccines, gene therapies, cell therapies, but really looking at the therapies that we feel are going to be important in the field of medicine for many years to come, and really looking to develop technologies to enable that type of development and enable it in the fastest way possible for our customers. That's really what we strive to do and what we continue to do. And that's the entire basis of where our platform technologies come from. So with that, I'll close for right now. I believe there are a couple questions, and I'll let Viet jump on and kind of run us through those. But any questions that you have, we're always happy to have, like I said, a conversation, high level or in depth, whichever you may like. But happy to help out however we can and answer any questions that we can.